Good morning, children. This is Perumal from Perumal Metric Higher Secondary School. Today we are going to see Chapter 5, Molecular Genetics, Part 3. Biospark, that is our channel. Children, today we are going to see the continuation of the chapter 5.11, tRNA, 5.12, Translation, Protein Synthesis, 5.12.1 Mechanism of Translation, 5.13 Regulation of Gene Expression. So in that 5.11 is deleted portion for this year. So we pass on to 5.12 Translation. That means protein synthesis. It is a process of polymerization of amino acid. It forms polypeptide chain. It is carried out by ribosomes that bind mRNAs and charged tRNAs. See here in the picture, you can easily find out what is the process going on. So the green color, that is the green color, this is a ribosome. It has two subunits that we study later. And here the mRNA, this is mRNA, this is a mRNA and charged tRNA. So what is the meaning of charged tRNA? This is a tRNA. When it carries one amino acid, then it is called charged tRNA. So now these are all assembled here, and one by one, as per the codon and anticodon pairing, the amino acids are lengthened. That is called polymerization. So it brings the polypeptide chain. So here you can see the polypeptide chain. This is called translation or protein synthesis. So this will take place in the 5' end. This is the 5' end of mRNA. Okay. So in the zip file also you can understand very easily how the polymerization take place. Now the mechanism. You know very well, all these processes are taking place in the ribosome. So it is called cellular factory. And in lower classes, children, you have studied this one. Ribosomes are called the protein factories. The protein synthesis takes place there. That's why we call like that. Now, we have two types of ribosomes. One is in prokaryotic and another one is eukaryotic. So in prokaryotic, the ribosomes are 70S. S that represents sedimentation efficient, which is expressed as Swartberg unit. The ribosome in prokaryote is smaller, but in the case of eukaryote, it is larger. Whatever the type, the ribosomes has two subunits, the larger and smaller subunits. In the case of prokaryote, the larger subunit is 50S, but in the case of eukaryote, it is 60S. And smaller subunit in prokaryote, it is 30S. In the case of eukaryote, it is 40S. Then in the ribosome, they have rRNAs. And there is a small change or small difference between prokaryote and eukaryote ribosomes. What are they? The 30S ribosome in the case of prokaryote, that has 16S rRNA. And the larger subunit that has 5S RNA and 23S RNA, then 31 ribosomal proteins. So this is about the prokaryotic ribosome. And here in eukaryotes, we have the smaller eukaryotic subunit consists of 18S RNA, then 33 ribosomal proteins. The larger subunit consists of 23 sRNA and 5S rRNA, then 31 ribosomal proteins. So these only the difference is in the presence of ribosomal proteins. Next one, reading frame. The reading frame is nothing but how the mRNA, the nucleotides are arranged, that we study now. It is used to determine which amino acid will be encoded by a gene. Open reading frame. Any sequence of DNA or RNA beginning with a start codon and which can be translated into a protein, then it is called open reading frame, ORF. Sometimes they may ask for expansion, expand ORF. So it is open reading frame. So when the codon is translated into a protein, then it is called ORF. Then untranslated region. mRNA also sometimes have additional sequences that are not translated. The start codon, it is AUG, begins the coding sequence and is read by a special tRNA that carries methionine. Methionine is the amino acid. Then ST sequence, Shine Dalgarno sequencer, that is the ribosomal binding site. Ribosomal binding site is SD sequence. This is about the reading frame. Then mechanism of translation or mechanism of the protein synthesis. So here we have three stages. One is initiation, elongation and termination. Here the initiation, the assembly of ribosomes on an mRNA molecule. Then elongation, repeated cycle of amino acid addition. Termination, release of the peptide chain. So all these three you can easily understand from the picture. So initiation, here there is assembly of ribosomes. This is a ribosome and here you have the mRNA. So there is assembly of the ribosomal molecules. Then elongation. So here what is happening? The amino acids are added one by one. So you will get a new protein. That is done by the charged tRNAs. Then at last termination. So when it reaches the stop codon, the polypeptide chains are released outside. This is about the brief introduction about mechanism of translation. Now we see one by one. First one, initiation complex. The initiation complex, it has three important components and three more the chemical factors. Now what are they? The ribosomal subunits, mRNA and tRNA. Then apart from that, there is three proteinase initiation factor, IF initiation factor, that is we call IF1, IF2, IF3. Then we have GTP, that is guanine triphosphate, and last one Mg2+. So these are all the initiation complex for translation. How it is taking place? Here the methionine, it is very, very important first amino acid incorporated into the protein. So here you can easily find out the three complex, the ribosomes. And next one, the tRNA with the methionine, that is initiation. Then the third one is mRNA. See here, this is a three complex. Then the interaction of the initiation complex. Now only we studied the initiation complex. What are they? Ribosomal subunits, mRNA and tRNA. So these are all the complex, initiation complex. And here it takes place in three steps. So first one, the initiation factor 3 binds to the 30S. 30S. 30S means it is a subunit of ribosome. Then it allows the 30S ribosome subunit to bind to the mRNA. So the blue color is the ribosome and this is a mRNA. So it binds. Later what is happening? Another initiation protein, that is IF2, then enhances the binding of charged formyl methionine tRNA to the small subunit in response to the AUG triplet codon. So here AUG triplet codon is there. Now the charged tRNA with the methionine will come and now it is translated. IF means initiation factor, then EFTU elongation factor thermo unstable, then EFTS elongation factor thermostable. Next, elongation. So the elongation is the second phase of translation. Ribosomes are assembled with the mRNA and charged tRNA molecules. So in this stage, you can easily find out they are assembled. The mRNA and charged tRNA molecules are assembled together. You can observe here in all the stage you can find it. Here the ribosomes has three important sites. Three sites. One is A site, that is amino acid site. Amino acid site. So here the first tRNA will be binding with this. Then peptidyl site, that is P site. So after that the amino acid one by one they are arranged by peptide bond. And last one, the E site, that is exit site. After that the tRNA will be released. Then elongation. Here we have steps. We see one by one. The charge initiator tRNA binds to the P site. So P site already we know it is a second site in the ribosome. Next one, elongation. We study one by one. Step 1, the charge initiator tRNA binds to the P site. So I hope you know the meaning for initiator tRNA, that is methionine tRNA. Then this step requires the correct transfer RNA and GTP and two proteins called elongation factor that now only we studied, EFTS, then EFTU. So these two proteins are required to complete the first step. Then in the second step, Peptidetransferase catalyzes the formation of peptide bond that links the two amino acids together. At the same time, the covalent bond between the amino acid and tRNA occupying the P site are hydrolyzed. That is, they are released or broken down. So to release this tRNA, the unchanged tRNA moves through E site and they move outside. Step 3, the entire mRNA, tRNA, amino acid 1, amino acid 2, this complex shift the direction of P site by a distance of 3 nucleotide. The step requires several elongation factor and the energy derived from hydrolysis of GTP. Step 4 results in the third triplet of mRNA to accept another charged tRNA into A site. Step 5 and 6, the sequence of elongation is repeated over and over. An addition of amino acid is added to growing polypeptide. Once the polypeptide chain is assembled, it emerges out from the base of the large unit. So in this picture also you can easily find out. So first in first step the same steps are explained very well in the diagram that is present in your textbook also. So refer the textbook. So here this is a subunit of ribosome. This is a large unit of ribosome. This is small subunit. Here we have A site, P site and E site. So here one by one the tRNAs are assembled as per the triplet. So first one, the red color, that is a methionine, F-met tRNA. Then the second charged tRNA will enter inside. And after that, what is happening? The peptide bond is formed between the added amino acid. Meanwhile, the first tRNA should be removed. So for that, there is a hydrolysis. And next, what is happening? Everything, this energy required for this are received from GTP. The energy is derived from the GTP. And later, another tRNA, charged tRNA, moves inside to the P site and transfer one amino acid. The peptide bonds are elongated. So many amino acids are added one by one and a peptide bond is formed. At last, when they reach the termination codon, the polypeptide chain is released. Termination. Termination is the third phase of the translation. Termination of protein synthesis occurs when one of the three codons, that is the stop codon, appears in the A site of the ribosome. So you know very well the stop codons. What are the stop codons? So when the stop codons reaches here in the A site, so it stops further translation. The terminal codon signals the action of GTP, dependent releasing factor, which cleaves the polypeptide chain from the terminal tRNA, releasing it from the translational complex. The tRNA is then released from the ribosome, which then dissociate into its subunit. Then 5.13, regulation of gene expression. The gene expression can be turned on and turned off. It was extensively studied in prokaryotes with the help of Escherichia coli. The cluster of genes with related functions are called operon. That is a 2-mark question, my dear children. What do you mean by operon? The cluster of genes. So here, all these genes, they perform related function. That's why we call operon. They usually transcribe single mRNA molecule. In Escherichia coli, nearly 260 genes are grouped into 75 different operons. Here, the operon consists of operator, promoter, regulator, and structural genes. So see here we have the operator O, then promoter P, then regulator I, then structural genes we have 3, X, Y, Z. Gene expression is regulated at transcriptional level or post-transcriptional level or translational level. The structure of the operon. Now we have studied what are the components in the operon. Now we study the structure. The unit of gene expression and regulation is done by the operon. It consists of one or more structural genes and an adjacent operator gene that controls transcriptional activity of structural gene. So this structural gene will be controlled or regulated by the operator genes. Now what are the components present in it? First one, structural gene, codes for protein, rRNA, tRNA recorded by the cell. Then promoter gene. These are all the signal sequence in DNA that initiate RNA synthesis. RNA polymerase binds to the promoter prior to the initiation of the transcription. The operators are present between the promoter and the structural gene. The operator gene is present in between the promoter and the structural gene. Then repressor protein, that is I, binds to the operator region of the operon. Now this is in general we have studied, and we have Lac operon or lactose operon. So here what are the components present? The same thing is present. Okay, don't worry. The same, all the components are present. One regulator gene I, promoter site P, then operator site O, 3 structural genes, namely Lac, Z, Y, and A. So these are all the components present in the Lac operon. Lac Z gene codes for beta galactosidase. Lac Y gene codes for permease, and A gene codes for transacetylase. This is a very, very important one more question, student. You have to remember what are the enzymes. So the structural genes, they produce the enzymes. So these 3 enzymes are important for the digestion of lactose. Now we study how the Lac operon functions. This was discovered by Jacob and Monod to explain the gene expression and regulation in Escherichia coli. In Lac operon, a polycistronic structural gene. Why we call polycistronic structural gene? Because we have 3 structural genes and they are doing related function. And these structural genes are regulated by common promoter and regulatory gene. The 3 enzymes produced by the lac operon structural genes are permease, beta galactosidase, then transacetylase. Now, what is the function of permease? It is needed for entry of lactose into the cell. So, lactose should enter inside the Escherichia coli. Then beta galactosidase, it brings about hydrolysis of lactose to glucose and galactose. So, on hydrolysis, you know very well, lactose gives glucose and galactose. And transacetylase, it transfers acetyl group from acetyl-CoA to beta galactosidase. So these are all the three enzymes produced by lac operon. Now we see how they are produced. Now two situations are given. One is when lactose is absent, and another situation lactose is present. So that's why in the previous slide we have studied the gene expression is on or off mode. Okay. Now lactose is absent and glucose is the source for energy. So in this situation, what is happening in the lac operon that we study now? When the cell is using its normal energy source from the glucose, what is happening? See here, now the I gene transcribes a repressor mRNA. So here an mRNA is produced from the regulatory gene. So this repressor protein binds to the operator. So what will happen if the operator is blocked or binds with the repressor? Because here the repressor is active. So the operon will not function properly. So beta galactosidase is not produced. And next situation, glucose is absent, lactose is available, so lac operon is on. So in this situation, what is happening? Lactose acts as an inducer. So when lactose is present, it acts as an inducer and interact with the repressor to inactivate. So lactose gives the energy. In the presence of lactose, the repressor is inactivated. The repressor is inactivated so that the repressor will not bind with the operator. It does not happen. So operator is free to function. This allows the RNA polymerase to bind to the promoter site and transcribe the operon to produce lac mRNA. Now the lac mRNA is produced. Okay. So this will enable the formation of all record enzymes needed for the lactose metabolism. So all the three enzymes are produced from the mRNA. This regulation of lac operon by the repressor is an example of negative control of transcription initiation. Lac operon. So both in your textbook in the same picture, both are given. When lactose is absent, glucose is present. So the repressor is active, so it binds to the operator, so no further action. But in the case of lactose present, the repressor is inactivated, so it will not bind to the operator. So the mRNA, lac mRNA is produced. So all the three enzymes are produced. It will act on the lactose. Thank you, children. We see in the next video.